Changing the genome of an organism means changing its characteristics. Humans have been genetically modifying plants and animals for thousands of years through selective breeding. Selective breeding involves choosing parents with desired particular characteristics to breed together and produce offspring with the desired characteristics. Humans have selectively bred plants and animals for thousands of years, including crop plants with better yields, ornamental plants with particular flower shapes and colors, farm animals that produce more, better quality meat or wool, dogs with particular physiques and temperaments suited to do jobs like herding sheep or hunting birds. Selective breeding aims to adapt an organism's characteristics in a way that is desirable to the humans that breed them. An organism's characteristics are partly determined by the combination of gene variants that are passed on from one generation to the next. For example, the children of tall parents may themselves be tall if they inherit a combination of tall gene variants. We can take advantage of this to selectively breed animals or plants, choosing parents with desired characteristics to produce offspring that have those desired characteristics. For example, if tall parents were bred together excluding short parents, the offspring should inherit tall gene variants that make them tall. Some of the offspring may even be taller than both of their parents, because they may inherit a combination of different tall gene variants from each parent, and together these make the offspring taller. With repeated selective breeding over multiple generations, this population will get taller and taller. After thousands of years, almost every single plant and animal around us is vastly different from its pre-domesticated state. Selective breeding and genetic engineering change an organism's genetic characteristics, but they are different processes. If humans have been changing genes for millennia, what makes a so-called genetically modified organism, or GMO, different? Selective breeding makes use of existing, naturally present gene variants in a species, and the natural process of breeding. Selective breeding is basically trial and error, hoping for lucky hits. Genetic engineering eliminates this factor. The desirable traits can be chosen, like making fruits grow bigger, immune to pests, and so on. With today's advances in genetic engineering techniques, the slow process of selective breeding can be sped up by incorporating specific new genes from one species into a completely unrelated species. Genetically modified organisms, or GMOs, have had their DNA artificially modified by humans using modern genetic manipulation techniques. Genetic engineering involves a direct change to an organism's genome in the laboratory. Gene variants made through genetic engineering can be passed from one generation to the next. How does genetic modification happen? There are several techniques that can be used to modify a genome, but the main one relies on bacteria. Bacteria are single-celled organisms that don't have a nucleus like our cells, but instead have one main circle of DNA, called the circular chromosome, and lots of tiny circles of DNA called plasmids, tiny bits of DNA that can contain one or several genes. Plasmids can be passed between bacteria easily, just like shaking hands. Let's go through the steps using the human insulin gene as an example. First, the gene for human insulin from a human cell is cut, using a restriction enzyme. The same enzyme is used to cut the circular bacterial plasmid open, and ligase is used to glue the insulin gene and the plasmid together. Then the modified plasmid is put back into a bacterial cell. Bacterial cells divide rapidly, and a little while later, we have millions of bacteria producing the human insulin protein and excreting it. The human insulin is filtered out and purified, and can then be used by people with diabetes. How simple is that for saving lives? Spider goats are also a great example of a genetically modified organism. They aren't as scary as they sound. The goats have had the spider silk gene inserted into their DNA. So when female goats lactate, they produce milk that contains spider silk. The spider silk is incredible. It's as strong as steel, but so light that a strand long enough to circle the earth would weigh less than 500 grams. There are thousands of uses for such a unique material. Scientists have used genetic modification to introduce genes that glow under UV light, such as from jellyfish, into other animals for medical research. This may seem totally useless, but being able to see certain cells in the dark has led to a lot of scientific discoveries. Glow-in-the-dark mice and fish have helped scientists make new breakthrough research into treating cancer, Parkinson's, and spinal cord injuries. Glow-in-the-dark cats have been used for HIV research. Crops can also be genetically modified to make them more pest-resistant, like the Bt maize. Maize plant was modified with genes of a bacterium Bacillus thuringiensis to synthesize delta protein, which is responsible for resistance against insects. As the insect eats the kernel, delta protein is activated in its gut that results in paralysis of the digestive system of the insect. The insect stops eating within a few hours and eventually dies. Another example is the Bt cotton, an insect-resistant variety of cotton. Genetically modified potatoes were modified using RNA interference to switch off genes to resist black spots, bruising, browning, and to produce low levels of amino acid asparagine that converts into acrylamide, a potential carcinogen, during frying of potatoes. Crops can also be genetically modified to have a nutritional benefit, like additional vitamin A, such as in golden rice. The rice variety Oryza sativa was modified with daffodil genes which synthesize more beta-carotene, which our body converts to vitamin A. Crops can also be genetically modified to have a longer shelf life. Flavor Saver tomatoes is the first commercially grown genetically modified food to be granted a license for human consumption. It is modified by adding an antisense gene to slow down the ripening process, thus preventing it from softening. Antisense gene interferes with the production of the enzyme polygalacturonase. This enzyme normally degrades pectin in the cell walls and results in the softening of the fruit, which makes them more susceptible to being damaged by fungal infections. Genetic modification can also be used to increase production. The Atlantic salmon was modified by a growth hormone regulating gene from a Pacific Chinook salmon. This gene increases the speed at which fish grows without affecting its size and other qualities in 16 to 18 months, rather than 3 years. GMOs are one of the most controversial areas of science. Why is that? Let's talk about some of the fears about GMOs. Let's start with one of the most common objections to GMOs. Gene flow, meaning GM crops could mix with traditional crops and introduce unwanted new characteristics into them. There is a method that might guarantee complete prevention, but is a big anti-GMO argument by itself: terminator seeds. The idea is that they could produce sterile plants, requiring farmers to buy new seeds every year. The very concept of this, however, caused a public outcry, stopping the technology being put to use. There are also cultural methods like buffer zones to keep unintentional crossing at a minimum. Is food that comes from GM crops different to food from non-GM crops? GM plants that are destined to be eaten are checked for possible dangers, and the results are evaluated by multiple agencies. After more than 30 years and thousands of studies, it is found out that eating GMO plants is no riskier than their non-GMO equivalent. But what about plants that have been engineered to be toxic, for example, Bt crops? A gene borrowed from the bacterium Bacillus thuringiensis lets engineered plants produce a protein that destroys the digestive system of specific insect pests. The plant makes its own pesticide and insects that eat it die. Pesticide sprays could be washed off, while the poison in Bt crops is inside the plant. Sounds alarming. Poison is just a question of different perspectives. What's harmless to one species might kill another. Coffee, for example, is a poison that kills insects, but is harmless to humans. Chocolate is dangerous for dogs, but a pleasure for humans. Bt crops produce a protein that is tailored to the specific design of the digestive tract of certain insects. It's completely harmless for us. Plants can also be engineered to be resistant to certain weed killers. This way, farmers can use these herbicides, killing the other plants competing for resources without harming the crop. The downside is, there is increased use of herbicides, good for the manufacturers, but bad news for the farmers who buy them, and the environment that absorbs them. This problem is actually more of modern agriculture and business practices of the huge corporations that control our food supply. This problem is not only valid, but also important. Agricultural practices must be changed to a more sustainable model. GMOs as a technology is an ally and not an enemy in the fight in helping save and protect nature and minimizing our impact on the environment. Let's look at some positive examples. Eggplant is an important crop in Bangladesh, but often, whole harvests are destroyed by pests. Farmers had to rely heavily on pesticides. Not only was this very expensive, farmers also frequently got sick. The introduction of a new GM eggplant in 2013 stopped this. The same Bt protein, an effective killer of insects, but harmless to humans, was engineered into them. This reduced insecticide use on eggplants by more than 80%. The health of farmers improved, and their income rose dramatically. In the 1990s, the papaya industry in Hawaii was under attack from the ring spot virus, which threatened to wipe out Hawaiian papaya. The solution was a papaya genetically modified to be resistant to the virus. Without it, the state's papaya industry would have collapsed. There is so much more we could do. Scientists are working on GMOs that could improve our diet. Plants that produce more or different nutrients, like fruit with higher antioxidant levels, that help to fight diseases, or rice with additional vitamins. On a larger scale, scientists are trying to engineer plants more resilient to climate change. These plants can better adapt to erratic weather and adverse soil conditions, making them resistant to droughts or floods. Scientists are working on crops that can absorb nitrogen from the air, like microbes. Nitrogen is a common fertilizer, but its buildup pollutes the groundwater and speeds up climate change. Plants that collect their own nitrogen could fix two problems at once: the overuse of fertilizers in the developed world, as well as the shortage of it in developing countries. Plants can be modified to become effective carbon collectors, like the American chestnut tree, to mitigate and actually reverse climate change. The world eats 11 million pounds of food every day. A UN estimate suggests the world needs 70% more by 2050. That amount of food could be grown by clearing more and more forests to create fields and pastures, and by using more pesticides. Or we find a way to do it on the existing agricultural land we've got right now, using more effective methods like GM crops. Intensifying farming instead of expanding it means GMOs could become the new organic. GMOs have the capability to not only significantly change agriculture, but to also reduce the effects of our own irresponsible behavior towards the environment. GMOs could be our most powerful ally to save our planet. With the tools we have today, our imagination is the limit.